Okay, I'm gonna just, give you some idea of some things that you need to study. It's going to be random. It's not going to go in order, so you can go back and you can listen to this, several times and write stuff down. First thing is you need to make sure that you know the functions of the dermis, the epidermis, the hypodermis. What are the layers of the epidermis? What are the layers of the dermis? And then the functions of those. You need to make sure that there's several questions on directional terms like superior, inferior, superficial, deep, proximal, distal. Just remember that proximal and distal is only used for the arms and the legs, and then superficial and deep are used for layers of the skin. So there's a couple of questions with directional terms. There is a question on a plane, whether it's a transverse, sagittal, or frontal plane. So look at those. Know your examples of macromolecules, like what is a monosaccharide, be able an example. What are the disaccharides, like sucrose, lactose, and maltose? What are the polysaccharides? Are going to be like starch, cellulose, glycogen is stored in our liver, in our muscles. Okay, so again, make sure that you know the monomers of your macromolecules. When it comes to a nucleic acid, the monomer is a nucleotide. The nucleic acids are DNA, which gives us our genetic material, and RNA, which is responsible for making proteins. Proteins monomers are going to be amino acids. You hook many amino acids together and you get a polypeptide or a protein. Remember, there is a primary structure, a secondary structure, a tertiary, and a quaternary structure of proteins. And lastly are the lipids. The lipids, what puts them all together is they do not dissolve in water. They are insoluble in water. And so different types of lipids are going to be our triglycerides, which saturated fats are going to be solid at room temperature, and they come from animals and they're bad for us. Unsaturated fats come from plants. They're liquid at room temperature and they are much better for us. We have the phospholipids with a polar phosphate head and a non-polar fatty acid, two fatty acid tails that are hydrophobic. They do not like water. And so our phospholipid bilayer is made of the phosphate and the fatty acid tails. And lastly, the steroids, as far as lipids go, the most important steroid is going to be cholesterol, which is a 4-fused ring structure. It's found in our phospholipid bilayer, and it is also the precursor to our sex hormones. Know your pH scale, just very in general, that 7 is neutral. Below 7, as you approach 0, is going to be more acidic, and then above 7, as you approach 14, it's going to become more basic. Another name for basic is alkaline. So make sure that you look at that. The bones of the skull, just your major bones, that your frontal bone is anterior, your occipital is going to be posterior, your parietals are to the top part of the skull, and the temporals are lateral to the other bones. Some of the main structures as far as bone markings go, like the sella turcica. What is its function? What is the function of the crista galli? What is the function of the foramen magnum, and what bone would it be found on? So some of this, just your major bone markings. You don't have to go back and study all of them, but just again, the major ones. Let's see, as far as, let's look some more things here. Give me a second. Go back and look at your membranes, your serous membranes. If it's visceral, it's stuck to the organ. If it's parietal, it lines the wall of the organ that the organ sits in, of the cavity that the organ sits in. And if it's a pleura, it is lungs. If it's pericardia, it's heart. And if it's peritoneum, then it is your gut or your abdomen. So make sure that you know where you would find like the visceral pleura and the parietal pleura, the visceral pericardium, the parietal pericardium, the visceral peritoneum, and the parietal peritoneum. Many people get out of the class and still don't know those. As far as, let's see, when you're talking about a muscle contraction, look over the chain of events. There's a picture I've showed you of a synaptic cleft. It's the axon terminal, and it's numbered. Make sure that you know the chain of events that goes as far as an action potential coming in, and then what leads to a muscle contraction. There are 4 or 5 questions on the functions of your systems, the body systems. So go back and look, which system is responsible for what. And so, like I said, a part of it will be matching. And then there's a couple of questions that, again, just state the function of that particular system. And you need to, choose the correct answer as far as that goes. Know all of the types of cells that are involved as far as mature cells of cartilage or chondrocytes, mature cells of bone or osteocytes, mature cells of fibers or fibrocytes, adipocytes or fat cells. Remember, your osteoblasts build bone, and osteoclast tear bone down or break down bone. And anytime you hear of a stem cell is going to be just a cell that can change into various other types of cells. When you talk about the chemistry, make sure that you know what the atomic number is, which is the number of protons, which equals the number of electrons, and that the mass number is going to be a decimal number. Remember, please, that if it is a 5 or above, you need to round it up to find out the number of neutrons. So you take the mass number and you subtract the atomic number from it, and you would get the number of neutrons. And you would be provided with both those numbers. You don't need a periodic table to find that. Remember that the ion that is positive, that has lost an electron, is the cation. The T, looks like a positive charge. And the one that has gained an electron and it's going to be negative is the anion. Know those. Enzymes, remember, are proteins. All enzymes are proteins, but not all proteins are enzymes. And then enzymes are, biological catalysts that speed up chemical reactions. They go into a reaction and they come back out, so they're not used up. So we don't have to go and take more, or extra enzymes because we're using them up, because they're not used up. They lower the activation energy that's needed to start chemical reactions. And again, they are going to speed up chemical reactions. As far as movement across a membrane, remember that passive transport, it requires no energy, and simple diffusion, filtration, osmosis, which is the movement of water, all of those are passive transport. Active transport requires energy. And if you bring it into the cell, it's endocytosis. If it is solid, it's called phagocytosis. If it is bringing in a liquid, it is called pinocytosis. And then taking something against the concentration gradient that's going to use energy, taking it out of the cell, that is exocytosis. So passive transport goes from high concentration to low. It goes down a gradient, like the boulder going down a hill. So it goes from high to low. Active transport goes from low to high, like pushing the boulder up the hill, going against the concentration gradient. So, therefore it's going to require energy. So look over those, and remember that as far as the movement of water goes, water moves from where it is greater to where it is less. So if you have a beaker of distilled water, you put a cell down in it, then water moves from where it is greater to where it is less. You've put a cell in a hypotonic environment, so water moves into the cell and the cell is going to swell up. If you put a cell in salt water, you've put that cell in a hypertonic environment. And so water moves from where it is greater to where it is less. Water is going to move out of the cell. The cell is going to shrivel up or crenate. And then again, if it were in the hypotonic, it's going to swell up, or it could lyse or split. And the cells would like to be in an isotonic solution, to where water is coming and going at evenly. It reaches equilibrium, so it does not gain nor lose water. Look over the movements as far as flexion and extension, dorsiflexion, plantar flexion, abduction, adduction, supination, pronation. Go back over those and make sure that you know, if you were given a scenario, when you would use that particular movement. Remember that actin and myosin are your myofilaments, and myosin is thick, actin is thin. Actin has the little active sites on it where myosin heads will attach. And when calcium is released, then the myosin heads can attach to the actin active sites, because we have those two proteins, tropomyosin, which is covering the active site when a muscle is not contracting, and you have troponin, which when calcium is released, it's going to cause the actin filaments to kind of shift so that the active site becomes open. The myosin heads can attach, and it gives us that sliding filament model of muscle contraction. Okay, when the actin and the myosin, when they move over one another, remember that they are not shortening at all. It's the sarcomere that is going to shorten. The structural unit of a skeletal muscle is a sarcomere, goes from Z disc to Z disc, and the actin and the myosin are not getting shorter, but the sarcomere is getting shorter. And so, it's going to might appear that they're getting shorter, but they're really not. I may have said some of these, I know I've said it before, but you may not have actually studied it, but we've talked about what muscle fatigue is. Not your muscles being tired. Remember, it's just when an action potential is coming down and the muscle just cannot contract. It will not contract. That's muscle fatigue. Look at what muscle tone is. What does muscle atrophy mean? Atrophy means pulling away from. So muscle atrophy is going to be when muscles pull away from the bone. Muscle strain is just, again, if you have overused a muscle or you have extended it beyond its stretchability. Look over your real names of your landmark names, like another name for your cheek is buccal, chin is mental, knee is patella, popliteal is behind the knee, calcaneus is your heel, your tarsals, metatarsals, your carpals, metacarpals. Just look over the real names for some of the common names that you may have. So going back to solutions, remember it's the solvent does the dissolving, and the solute is what is dissolved. So water is the solvent and Kool-Aid is the solute. If it can dissolve in water, then again, it's going to be a solution. And you're not going to see any particles. They are going to be evenly dispersed throughout. You can shine a light through it and it's going to go through it. A colloid has larger particles. You will not be able to shine a light through it like fog. It will scatter. And then a suspension has very large particles that will settle to the bottom, just like Italian dressing or a snow globe of sorts, where you would have to shake it up, and everything starts floating around again, but let it stand long enough and it will eventually settle to the bottom. The most abundant protein in the body is collagen. Collagen is the most abundant protein. And as far as tissues go, remember that connective tissue is the most abundant of the four tissues. You have epithelial, connective, muscle, and nervous tissue. And then out of the connective tissues, collagen is going to be a fibrous a fiber, a very thick fiber of dense regular connective tissue. The most abundant compound in us, remember that we are made up of CHON, carbon, hydrogen, oxygen, and nitrogen, and the most abundant molecule is a molecule and a compound. Remember, a molecule has two or more atoms joined together, and a compound is two or more different atoms joined together. So water, H2O, is a molecule. It's two or more atoms joined together, but it's also a compound. It's two or more different atoms joined together. So the most abundant compound in our cells is water. Know where your organs are located so that you can use those directional terms. Also, as far as quadrants go, remember that the appendix it's going to be in the lower right quadrant. Your liver and gallbladder, most of the liver is in the upper right quadrant. Your stomach is over in the spleen area is in the upper left quadrant. And then your large intestine kind of spans all of the quadrants. It's the ascending colon, the transverse colon, the descending colon. And so look at where your organs are as to what quadrant they would be found in. The three kinds of muscles. Skeletal muscle is, of course, under voluntary control. It's conscious. You know whether you were going to move a skeletal muscle or not. It's striated. It's got several nuclei because a skeletal muscle cell or fiber is very cylindrical. It's very long, so it has more than one nucleus. Cardiac muscle is also striated, but it's got what we call intercalated discs, these little thick bands kind of. And remember that cardiac muscle is involuntary. You have no control over it, and it's only found in the heart. And smooth muscle, if it's not attached to bone and it is not in the heart, then it is going to be smooth muscle that is spindle shaped. It has no striations at all. And so it will line all of our hollow organs and pretty much all of the sphincters and anything, as I said, other than your heart and muscles that are attached to your bone, everything, all of those will be smooth muscles. Okay. Remember with the epidermis going from outside in, come let's get sunburned. So the cornea is, the stratum corneum is the outermost layer that gets sloughed off, is completely dead. Remember, if it's the palms of the hands and the soles of the feet, you're going to have that extra layer. So the lucidum would be, squeezed in there. Then you have the granulosum. Then you have the spinosum, and then you have the basal layer where mitosis is going on. That's where you're going to find melanocytes, or in that basal layer of the epidermis. That's the area that is undergoing mitosis. It's living, and it's undergoing mitosis. As far as muscles and what their job is, remember that the prime mover, otherwise known as the agonist, is the muscle that does a particular movement. A synergist is going to be a muscle that helps the prime mover. And then you have an antagonist that does the opposite movement of the prime mover or the agonist. So look over those. know, like, the just the different terminology as far as the if it's epi, whether it is epimysium or epineurium. Remember, that's to the outside. The perimysium or the perineurium is going to surround a fascicle, which is either going to be bundles of muscle fibers or bundles of nerve fibers or bundles of axons. That's what a nerve is. And then the endomysium or the endoneurium is going to be around, individual either muscle fibers or an axon of a neuron. As far as cellular respiration goes, just very basic, that glycolysis takes place with or without oxygen in the cytoplasm. Only 2 ATPs are made. If oxygen is present, then it will cross over into the pyruvic acid. Remember, will be converted to acetyl-CoA, and it can cross the mitochondrial membrane into the matrix where the citric acid cycle will take place. 2 turns of the cycle. It takes 2 turns of the cycle because there are 2 pyruvic acids that are made. Remember, for 2 turns, it's going to be 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. And then most of the ATP is made in what we call the electron transport chain, the ETC, which is in that inner mitochondrial membrane called the cristae, for a net total of, 36 to 38 ATPs. But your book, I think it said 32 ATPs. So again, if you see 32, then choose that. But if it won't be 32, 36, and 38 in one question. It's just going to be one of them. So just, make sure that you look at that. Know the pH of blood. Know that a 0.9 saline solution is going to keep a red blood cell isotonic. And remember, a 5% dextrose or glucose solution would help maintain homeostasis for that red blood cell. It's not going to cause it to swell or to crenate. As far as some of the muscles go, just very, very common muscles, like what are the 4 quadricep muscles? Which of the following is or is not a hamstring? Which is not a rotator cuff muscle? What is the smiling muscle? They're very general as far as I picked up what the muscles do. Okay. Remember again that osmosis is the movement of water going from an area of high concentration to low concentration. Go back and just look at, one place pretty much that your tissues can be found. Your epithelial tissues, if it's, remember, simple is 1 cell layer, stratified is many. And then it can be squamous, which is flat, cuboidal is cube-shaped, and columnar is column-shaped. So in the alveolar sacs of our lungs, in the capillaries, our blood vessels, where you want 1 cell layer thick, then you're going to have your simple squamous. All of the ducts, DUCT, like in the urinary system, those will be simple cuboidal. Remember that simple columnar will line your digestive tract. It'll have microvilli on the top that increases the surface area. Remember, cilia causes the movement of some kind of substance across the surface of the cell. But what lines our respiratory tract will be that ciliated, pseudostratified columnar epithelial tissue that lines our respiratory tract. All right, let's just give me a second. I've already mentioned the movements as far as abduction, adduction, flexion, dorsiflexion, all of those. Okay. In general, just know in general, a function of the different parts of the brain. Remember, the hypothalamus, it controls our temperature, our hunger center, our thirst center, our drives, like our sex drive. It helps to maintain homeostasis. But the medulla oblongata is the one that controls the part that controls our vital signs, like our blood pressure, our heart rate, our respiratory rate. The medulla does that. Remember that the thalamus is a relay station where everything comes in and it sends it to its appropriate place. Remember that the cerebellum is for balance and equilibrium. Remember that the cerebral hemispheres, as far as your frontal lobe goes, that is where your higher order thinking is, in the frontal lobe. And you have that motor cortex that is in the frontal lobe. You have the somatosensory cortex that is in the parietal, occipital, and temporal lobe, for all of our special senses. And so just, again, know in general that the occipital bone, of course, is for sight. The temporal is for hearing and smelling. Gustatory is, kind of in there too, but the parietal is kind of a somatosensory association area going on there. Your paranasal sinuses, the function of them, they make the skull lightweight. They allow for the air to be moistened as we breathe it in. They allow for resonance as far as us talking goes. And remember that we have an ethmoid sinus, a sphenoid sinus, a frontal sinus, and a maxilla sinus, our upper jaw, not your mandible, not the lower jaw. Okay, so know kind of what the sinuses do and what are our sinuses. Know the parts of a reflex arc. We just have had that. Know that it comes from a receptor. If it's somatic, it's in the skin, comes from a receptor. It goes up a sensory neuron or afferent neuron. It will be interpreted at the inner neuron or an integration center. It is sent back down a motor neuron or an efferent neuron to an effector, which is a muscle or a gland. Okay, so I just said that, of course, the sensory brings impulses to the central nervous system, and motor will take messages away from your brain and your spinal cord. Look over your neuroglial cells. Know that the ones for the central nervous system are going to be astrocytes, which are the most numerous. Your microglial help with the immune response, because remember, our immune system doesn't really work in the brain. Your ependymal cells will line any place that has cerebral spinal fluid. They have cilia on top. Then the oligodendrocytes are what makes the myelin sheath in the brain and the spinal cord. And then in the peripheral nervous system, we have the satellite cells, which are like astrocytes, and we have the Schwann cells, which are like the oligodendrocytes. It provides myelin, which insulates the axon. I mentioned earlier that a mature bone cell is an osteocyte. Remember, it's found in a little lacunae, a little bubble, and compact bone has concentric, what we call lamellae, with a central canal running down, that's going to have the blood vessels and the nerves. And so the osteocytes have little canaliculi, these little extensions, finger-like extensions that come off of it so that nutrients can pass from that central canal all the way out to that outermost lamellae, those cells that are on that last ring. Remember that the chondrocytes are mature cartilage cells, and they are also in a lacunae. The most numerous or the most abundant cartilage is hyaline cartilage. Again, look over where you would find hyaline cartilage. Then you have elastic cartilage that is in the top of your ear and the epiglottis. And then you have the fibrocartilage which is in your intervertebral discs between your vertebra, and that symphysis pubis that is between your ischium pubis bones, the right and left. And so again, look at that. One of the very first things that we learned were levels of organization. So it goes from your atoms and molecules that end up making compounds. And so that's kind of a chemical part to it. And then you've got your organelles, which will make up cells. Like cells make up tissues, tissues make up organs, organs make up organ systems, and organ systems make up the organism. That is going, make sure you read the question. Is it going from the least complex to the most, or is it going from the most to the least? If it's most, you're going from the organism first. If it's going from the least, you're going from the chemical aspect of it, which is going to be the least. Just some general parts of the eye, as far as the cornea and the sclera. They do not have blood vessels. You have the lens there that can change its shape to accommodate our distance, whether it's close up or far away. Go back over and look at the passageway of light through the eye. You have that aqueous chamber or aqueous chamber with a fluid. Remember, it is circulated through. It is made, and then it will be drained through that scleral venous sinus, and it gets into the bloodstream, but then it's constantly being replenished in that anterior chamber. It's not stagnant. Then you have that posterior chamber, which is the vitreous humor. You have that choroid coat that runs around. Well, the sclera, actually, the white of the eye runs all the way around to the optic nerve. The choroid coat is the next layer in. The retina is the innermost layer, where you're going to find our photoreceptors, which are rods that allow us to see in dim light, and in shades of gray and black. And our cones will allow us to see in color, and know that, that we have the blind spot. The fovea centralis is where the optic nerve is actually leaving the back of the eye. So when, they look into your eye, when an eye doctor, ophthalmologist looks into your eye, or optometrist looks into your eye, they can see all the way back to the retina. It goes all the way through. They're checking the pressure of the eye to make sure that it is not under too much pressure. But then, they can see where the optic nerve leaves the back of the eyeball, but then they can't see past that. So they can't see past the optic nerve, and where it crisscrosses at the optic chiasma, that's in the on the brain. And so they're not going to be able to see that. So just parts of the eye. And the same thing with the ear, the path of sound through the ear, starting with you can call it the auricle or the pinna, going all the way to the semicircular canals and the cochlear. Okay. Remember, parts of a long bone, you have the shaft is the diaphysis. The ends are the epiphyses. If it's closest to the body, it's the proximal epiphysis. If it's further away from your trunk, it's the distal epiphysis. Remember, it's an epiphyseal plate. If there is still cartilage present, it's an epiphyseal line if it has ossified. Going down the middle of a long bone is a medullary cavity that is filled with yellow marrow. Okay. We just had this. What are some characteristics of the sympathetic and the parasympathetic division? There's only, one or two questions on that. But again, if you did not learn it for the test, then go back and remember that your sympathetic is your fight or flight. Okay. And look at what characteristics characterize, the sympathetic division, and then the opposite would be the parasympathetic. Okay. As far as protein synthesis goes, just basically know where they take place. Transcription takes place in the nucleus, and it is the making of mRNA from DNA, a DNA template, Remember that DNA has ATGC. There's two questions. One is going to say, give me the complementary strand of DNA. So your answer cannot have a U in it. It must have a T in it. And then there's the one that says the complementary strand of messenger RNA from DNA. That has to have a U in it. So RNA is AUGC. So don't miss that. Transcription taking place in the nucleus, and it is the making of messenger RNA. And translation is taking place in the cytoplasm with the transfer RNA. Remember, it's got an anticodon on one end, and it's got an amino acid on the other end. The anticodon hooks into the codon on messenger RNA. The start codon is AUG. It codes for MET, methionine, and the three stop codons are UGA, UAG, and UAA. That's pretty much it as far as protein synthesis takes place at the ribosome. I didn't say this. There are probably 5 questions, a couple of them are matching too, as to the functions of the cellular organelles. Okay. So again, just in general, look over, the functions of the organelles. There were 2 kinds of bone ossification. We had endochondral bone ossification, where you have a cartilage mold of a bone. And remember, you have that primary ossification center that starts to eat away at the medullary cavity. You have the secondary ossification center is at the epiphyses, until finally you end up with a long bone, and you have the articular cartilage will be on the ends of the long bone, and you have that epiphyseal plate that still will be cartilage until it ossifies. Every long bone is made by endochondral ossification. Intramembranous ossification occurs at the flat bones of the skull, and so that's what leaves behind the sutures. Okay. The 3 kinds of burns. First degree burn only burns the epidermis. A second degree burn burns the epidermis and part of the dermis, and it's a second degree burn that you would find a blister. And a third degree burn or full thickness burn burns the epidermis, the dermis, and the hypodermis. And that is the one that usually needs skin grafting. Okay. People get this mixed up. Still, by the final, you still don't know your glands. Okay. Sudoriferous glands are sweat glands. We have 2 kinds of sweat glands. We have eccrine, which go all the way to the surface to a pore. And that is, they're activated when we heat up. When our temperature increases, then it's the eccrine sweat glands that are opened up. Apocrine glands open into a hair follicle. And so apocrine glands are going to be found in the armpit, in the axillary area, and the groin area. And those are the ones that are activated at puberty, and they're the ones that have a smell. Sudoriferous glands. A sebaceous gland is an oil gland. So a sebaceous gland is associated with every hair follicle, and an apocrine gland is associated with every hair follicle. The ecrines are not. And remember that it's the little erector pili muscle that when you get cold and you shiver, it causes your hair to stand straight up. Okay. I told you earlier about the ependymal cells that have the cilia, and wherever there is cerebral spinal fluid, that's where they're going to be found. So cerebral spinal fluid is found in the subarachnoid space. It's found in the ventricles of the brain. It is found in the central canal of the spinal cord. So again, you've got the pia mater and the arachnoid mater. You have between those two is the subarachnoid space, sub meaning below the arachnoid. And so the dura mater is going to be the furthest one out, closest to the surface. Then you have the arachnoid mater. Then you have the subarachnoid space. Then you have the pia mater that is stuck directly to the brain. Okay. I think I've already said where we find our sharpest vision is our fovea centralis. Just look at, terms like blind spot, optic disc, fovea centralis, maculae or maculute. Let's see. Structure of a muscle. I kind of touched on that. You have the whole muscle. And then it is broken down into fascicles, which are bundles of muscle fibers. A muscle fiber is a muscle cell. Then you have the myofibrils, and then myofibrils are made up of myofilaments, which are the two are actin and myosin. Okay. I've already said, make sure you know where your organs are located. It's not going to, it's not going to use any abdominopelvic answers. It's going to either be in the abdomen, it's going to be in the pelvic cavity, it's going to be in the thoracic cavity. It's going to be, if it says which is the following is in the dorsal cavity, remember your brain is in the cranial cavity, and then your spinal cord is in the vertebral or the spinal cavity. Your thoracic cavity would have your pericardial cavity. With your heart, it'll have your pleural cavity. For your lungs, the diaphragm separates it from the abdominal cavity where all of your abdominal organs are. And then the pelvic cavity will have your urinary bladder. Women, it'll have the uterus, and it will have our other reproductive organs, as far as male and female internal reproductive organs go, would be that pelvic cavity. Okay. Know the reactions that a catabolic reaction, the way I remember it is, it's kind of silly, but cats don't like water. And so a catabolic reaction, another name for it is hydrolysis. Hydro means water, lyse means to split. So you've got the whole large macromolecule, the whole protein is broken down by putting water in, it splits apart, and it will be broken down into amino acids. When you break a bond, energy is released. Dehydration synthesis. Dehydrate means to take water out. That's another name. Another name is an anabolic reaction. So now you've got the little parts, the little amino acids that you want to hook together to make a protein. So you take water out and you hook them together. So look over those reactions, how they're named, and the different names for them. Let's see here. I think I've already said about a codon. A codon, remember, codes for one amino acid. If it is UUU, I think that's phenylalanine. But remember that because there are just 20 amino acids, each amino acid could have more than one codon that codes for it. So codons on messenger RNA are coding for an amino acid. Okay. Look over the words. What does it mean to be polarized? Polarized is at rest. Resting membrane potential, -70 millivolts. It is negative on the inside and positive on the outside. When an action potential comes along and the sodium channels are open, then it becomes depolarized. So whether it's going to be positive on the inside and negative on the outside. And as the sodium channels close and then the potassium channels open, then it will become repolarized to where it's once again going to be negative on the inside and positive on the outside. Look over those words. As far as what do they mean? Hyperpolarization, look over what that means. What do tendons bind to? Tendons hook a bone to a muscle. Ligaments hook bone to bone. Remember that one of the thickest tendons that we've got is the calcaneal tendon or the Achilles tendon, whichever one you want to call it. And then we have many, many ligaments, all throughout the body. If a bone is growing in thickness, okay, if it's going to grow in thickness, we may not have it does grow in length, but again, our bones will stop growing in length once they have become ossified. But our bones are constantly restructured and rebuilt and broken down all throughout our life. And so apositional growth is when bones it'll change the thickness of it. They grow in thickness, and that would be apositional growth. Just give me a second. Questions about which of the following is not associated with the appendicular skeleton. Appendicular are the arms, the legs, and associated girdles. The axial skeleton is the head, neck, trunk. So it's the skull, the vertebral column, and the rib cage. That's an axial skeleton. The appendicular skeletons are the bones in the arms, bones in the legs, and then your pectoral girdle, which is your scapula and your clavicle, and your pelvic girdle, which is your ilium, ischium, and pubis bone. Okay, so make sure that's an easy question. Make sure you don't miss, which one of the following is not in, one of those branches there. Remember that we are studying anatomy, which is the study of the body parts, or the parts that are associated with the systems and the structure of them. And physiology deals with their function. Remember, anatomy and physiology go hand in hand, that they correlate with each other, that a lot of times that the anatomy of something helps determine its function, and vice versa. If it has a specific function, then it might need to have a certain makeup or a certain shape so that it can carry out that function. Look over your reflexes, the different terms that we use for reflexes, whether it's a flexor reflex, if it's blinking, if it's the withdrawal reflex. Just look over those and what's involved with those. Remember, I did say that about the atomic number and the atomic mass. The number of protons is the atomic number, which equals the number of electrons. Remember that the rule is 2 electrons can go in the innermost shell, up to 8 can go in the second shell, and up to 8 can go in the third. So if it has atomic number of 13, okay, 13 is going to be, you have 2 in the first, you have 8 in the second, that's 10, and you have, we got to get to 13, we have 3 in that third shell. So we have 3 valence electrons. Valence electrons are the electrons in the outermost shell. I think that was sodium that I just did. Let's see. All right, just give me a second. I've already told you about the monomers of the different macromolecules, that phospholipids are made up of glycerol and a phosphate head and glycerol and 2 fatty acids. A triglyceride is glycerol and 3 fatty acids, your saturated and unsaturated fats. Know that your thumb, remember, we didn't really think about it, but your thumb is called your pollex and your big toe is called your hallux. Okay. Look over your joints. Which of the following is an example of a synovial joint? Remember, you've got your hinge joints, your saddle, your pivot joints, your ball and socket, your condylar joints. Those are all synovial joints that have a cavity. Okay. We have the fibrous joints, and we have the immovable joints like the sutures. So look back over your joints and examples of them. Let's see. Just give me a minute. Look at the shapes that muscles can be like. If it's circular, remember we have the orbicularis, the oculi, and the oris are circular. We have the pinnates, which we have the unipinnate, and it's going to be attached to a tendon on one side. The bipinnates are attached to the tendon on two sides, and multipinnate is when you have several almost bipinnates kind of put together. The deltoid is an example of multipinnate. You have the fusiform, which is going to be the spindle shaped. You have the fan shaped, like the pectoralis muscle and the temporal, or convergent. Look over those. You should know that we've got 12 pairs of cranial nerves and 31 pairs of spinal nerves. Do you know that? Would you be able to pick out an organic compound? Remember, organic compounds have carbon, of course, except carbon dioxide is not organic. Carbon monoxide, the bicarbonate ion, is not organic. But our big organic molecules are carbohydrates. And remember, any sugar is a carbohydrate, whether they are monosaccharides, disaccharides, or polysaccharides. Your lipids are organic. Your proteins are organic, and the two nucleic acids, DNA and RNA, and ATP has we don't really think of it as a nucleic acid, but it is first cousins to a nucleic acid. Those are all organic molecules. Inorganics are going to be sodium chloride, potassium bromide. Your salts, water is inorganic. Any of your gases are inorganic. Any of the metals, gold or your lithium, aluminum, all of those are inorganic. They never have lived. Okay. All right, just give me a second here. Already talked about glands, whether they are sebaceous or they are sudoriferous glands. Just give me a sec. I'm just looking. I've already talked about muscles, which one are voluntary and involuntary. Okay, what is a buffer? Remember that a natural buffer is the sodium bicarbonate ion, but we don't want to get too acidic. The closer to one we get, the more acidic it is. There are places on us that like to be acidic, like our stomach acid with a pH of about 1. But we don't want to get too basic either, or too alkaline. So we have a buffer that helps stabilize our pH so that it doesn't go to extremes. All right, remember that the olfactory is nerve number 1, optic is nerve number 2, oculomotor is 3. And go back over and look at the numbers of your cranial nerves. Remember that nerve number 10, vagus, is the only nerve that extends beyond the brain. It innervates all of our major organs. The vagus nerve, number 10, is the one that will go to all of our major organs, regulating, our abdominal viscera, in there. What about bonds? If you share electrons, you are covalent, which is the strongest bond. If you gain or lose electrons, you will form an ionic bond. Ionic bonds can be broken, and so when they're broken, they will break down into an anion, which will be negative, and a cation, which will be positive. And then hydrogen bonds are, again, the weakest of the bonds, but covalent bonds are sharing of electrons, and they are the strongest of the bonds. As far as the cell cycle goes, remember you have interphase, and it includes G1. S is synthesis. That's when your DNA makes a copy of itself. and you have G2. So your cell is getting ready to divide. And then you have mitosis, prophase, metaphase, anaphase, telophase. And then you have the splitting of the cytoplasm, which is cytokinesis. Know just in general what is happening in each one of the stages of mitosis. In prophase, the nuclear envelope disappears, the nucleolus disappears. The chromosomes are visible. They have become shorter and thicker so that you can actually see them. They're not long and stringy chromatin. The centrioles, the pairs, there'll be 2 pairs of centrioles will move to opposite ends of the cell. Then your chromosomes will line up down the middle, metaphase, middle. The chromatids, one arm of the chromosome, sister chromatids, they will pull apart during anaphase, apart in anaphase, A and A. And then telophase is the opposite of prophase. The nuclear envelope will reappear. The nucleolus will reappear. The centrioles will move back to their place right outside of the nucleus. And your chromosomes will kind of be more dispersed and they will become stringy, and we call that chromatin. So your genetic material is chromatin, but when your cells start to divide, then they condense and get shorter and thicker, and we call them chromosomes. Look over what we need to survive. You know, we need, remember there were things that for us to live, like movement and reproduction and growth, and there was a whole list of them. But then there was 5 things that humans needed for survival, like we would die without it. And so definitely water and oxygen and nutrients. Remember, we have to have appropriate atmospheric pressure so that we don't crush. And so again, we have to maintain, you know, our normal body temperature because we are endothermic. We're not exothermic or endotherms like amphibians or reptiles, where their temperature can fluctuate. Ours has to stay at a nice even about 98.6 degrees ish. Okay, so look at those, what we need to survive or what we have to have for survival. Look at your taste again. But just, you know, go back and look at what causes us to be able to taste bitter. Again, that's going to be, if it's bitter, it's going to be your, your alkaline structures. If it's sour, it's acids. Umami is going to be your amino acids, glutamate and aspartate. Salty is metal ions like sodium chloride, sodium bromide, potassium chloride. Those are metal ions. And then sweet, of course, is going to be your organic carbohydrates, your organic substances like sugars. So go back and look at that. And so if you'll take this, and you might have to listen to it a couple of times and write some stuff down, I feel like you should be, you know, pretty prepared for the test. Like I said, it's very general, and you know, I feel like it's 150 questions. I think we've got almost 2 hours to take it, but I feel sure that people will finish in an hour. It's that easy. So, you know, be looking over everything. Again, I'm sorry for posting this so late. I just have had a lot going on this weekend, but this at least will help you. And if you do need me before your test, Monday, people, before Monday night, I will be in my office. My 117, they're taking their exam in the morning at 8 o'clock. And so I'll be back in my office, you know, around 10 or 10:30, but I'll be there for the rest of the day. So Monday night, people, if you need me, Tuesday night, people, you know, please make sure that if you need me, by all means, call me or email me or come by and see me. And then Tuesday, face-to-face, people, again, if y'all do need me, I am around tomorrow. So if there is nothing else, then hopefully this did help. And I wish all of you the best of luck. All of you you'll do great.